Snakes lost their limbs over 100 million years ago, some 35 million years before T-Rex roamed the Earth. But scientists have struggled to identify the genetic changes involved in this transition. Now, a new study has described a stretch of DNA involved in limb formation that is mutated in snakes, which could shed light on the slippery matter. Scientists from an array of Californian institutions and the University of Basel in Switzerland inserted the snake DNA into mice and discovered the animals developed truncated limbs, suggesting a critical stretch of DNA lost its ability to support limb growth during snake evolution. This is one of many components of the DNA instructions needed for making limbs in humans and, essentially, all other legged vertebrates, said Axel Weissel, a geneticist at the Lawrence Berkeley National Laboratory in California and senior author on the paper. In snakes, it's broken. It's probably one of several evolutionary steps that occurred in snakes, which, unlike most mammals and reptiles, can no longer form limbs. Modern serpents have undergone one of the most dramatic body plan changes in the evolution of vertebrates. Dr. Weissel and his team decided to look at the published snake genomes of basal snakes such as the boa and python, which have vestigial legs, tiny bones buried in the muscles, as well as advanced snakes such as the viper and cobra that don't have the bones, in order to study the molecular roots of the adaptation. Within these genomes, they focused specifically on a gene called sonic hedgehog, which is involved in many developmental processes, including limb formation. The researchers examined one of the gene regulators, a stretch of DNA called the zone of polarizing activity regulatory sequence, that was present but had mutated in snakes. To determine the consequences of these mutations, they used a genome editing method called CRISPR to insert the ZRS from other vertebrates into mice. They found that when the sequence from mammals was inserted, it led to the development of normal mouse limbs, as described in the Cell Journal. Even when they inserted the ZRS from fish, whose fins are structurally very different from limbs, the mice developed normal limbs. However, when the researchers replaced the mouse CRS with the python or cobra version, the mice went on to develop severely truncated forelimbs and indolimbs, suggesting this stretch of DNA was responsible for the change in snakes. To ensure the snake CRS was responsible in losing the serpent's limbs during their evolution, the researchers took a closer look at the evolutionary history of individual sequence changes. By comparing the genomes of snakes and other vertebrates, they identified one particularly suspicious 17 base pair deletion that only occurred in snakes. This deletion removed a stretch of the ZRS that has a key role in regulating this gene in legged animals. They then turned back the evolutionary clock by restoring the missing 17 base pairs in an artificially created hybrid version of the python ZRS and tested the edited DNA in mice. The team found mice that carried this evolutionarily resurrected ZRS in their genome developed normal legs. While the mystery now appears to have been solved, Dr. Weissel said the evolutionary events that led to snakes slithering along without legs were probably more complex than just the one deletion. There's likely some redundancy built into the mouse CRS, he said. A few of the other mutations in the snake CRS probably also played a role in its loss of function during evolution. Of course, snakes aren't the only vertebrate animal that lack arms and legs. Some lizards, eels and other fish and marine mammals have also shed their limbs over millennia, although likely via a different evolutionary process. Loss of limbs has occurred multiple times independently during animal evolution, and it's safe to assume that mutations affecting other genes were involved. Dr. Weissel said, "It's a complex problem, but with the introduction of genome editing tools, we can finally start tying specific DNA changes to alterations in body shape more systematically."